Hello everyone. In this video I would be discussing about the 8 important points from the topic of citric acid cycle. The first point that you need to remember is that there is no net removal of oxaloacetate in the citric acid cycle. In my previous video I had discussed that citric acid cycle begins with the condensation of acetyl-CoA with oxaloacetate to form citrate, which is a 6-carbon molecule. And in that video I had explained that in the last step of citric acid cycle, oxaloacetate molecule is regenerated. So in short, there is no net removal of oxaloacetate in the citric acid cycle. The next point that you need to remember is that in one round of citric acid cycle, 3 molecules of NADH are produced and 1 molecule of FADH is produced. Also, 1 molecule of GTP is also produced. Next important point that we need to remember is that in one round of citric acid cycle, total of 10 ATPs are produced. I repeat, in one round of citric acid cycle, total of 10 ATPs are produced. It's because in one round of citric acid cycle, 3 molecules of NADH are produced and 1 molecule of NADH gives 2.5 ATP. So 3 molecules of NADH will give 7.5 ATP. Also, in one round of citric acid cycle, 1 molecule of FADH is produced and 1 molecule of FADH gives 1.5 ATP. So 3 NADH are giving 7.5 ATP and 1 FADH is giving 1.5 ATP, which makes total of 9 ATP. Also, in the citric acid cycle, there is one step which involves substrate level phosphorylation, the step in which GTP is produced. So that 1 GTP is equivalent to 1 ATP. So adding that 2, we get 10 ATPs in one round of citric acid cycle. Now we move on to our next point, which involves oxidative decarboxylation. Oxidative decarboxylation reactions are those reactions which involve removal of carbon dioxide molecule as well as oxidation. And there are 2 steps in the citric acid cycle which involve oxidative decarboxylation, and those steps are catalyzed by the enzymes isocitrate dehydrogenase and alpha-ketoglutarate dehydrogenase. Next point discusses about the substrate level phosphorylation. There is only one reaction in the citric acid cycle which involves substrate level phosphorylation, and that step is catalyzed by succinyl-CoA synthetase. Succinyl-CoA synthetase enzyme converts succinyl-CoA to succinate, and in this process a molecule of GTP is produced. So this reaction is example of substrate level phosphorylation. In the previous slide we had discussed that in citric acid cycle 3 molecules of NADH are produced and 1 molecule of FADH is produced. And succinate dehydrogenase is the enzyme which catalyzes the reaction in which FADH is produced. Succinate dehydrogenase converts succinate to fumarate, and in this process a molecule of FAD is converted into FADH. Thus, succinate dehydrogenase is the enzyme which catalyzes the reaction of citric acid cycle which involves FADH production. Succinate dehydrogenase enzyme is not only the part of citric acid cycle, it is also part of the electron transport chain. As discussed earlier, succinate dehydrogenase converts succinate to fumarate, and succinate dehydrogenase enzyme is inhibited by malonate. Malonate is a competitive inhibitor of the enzyme succinate dehydrogenase.